Hi guys, so we are going to take some flip video notes on genetics. For some of you this might be a refresher, others it might be completely new. So we are going to learn about genetics, traits, and alleles to help us with the project that we have coming up in GTT. So what is genetics? Heredity is the process in which traits are passed from parents to offspring. And the scientific study of heredity is called genetics. Genetics is the study of heredity and how traits are passed on. And genes can come from the chromosomes, which can be found in the eggs, which is the female, and the sperm from the male. Each parent, especially in humans, contribute half of their pairs of 23 chromosomes to their child, 22 of them being autosomal chromosomes, and one of them being the sex chromosome. So every parent, regardless, gives 50% of their DNA. The moms give 50% of all 23 pairs, and the dads give 50% of all 23 pairs in the sperm. When the two come together, that gives the offspring 46 chromosomes, 23 from the mom, 23 from the dad, to create them, the new offspring. So here is a great picture. I do actually want you to draw this in your notebook because it's going to be very helpful. Everything that makes us who we are is DNA. It is the blueprints that tells us to have brown eyes, blue hair, red hair, straight hair, curly hair, long eyelashes, short eyelashes. Everything that we are naturally comes from the blueprints of our DNA. So DNA is in the shape of a double helix. It then wraps extremely tight into, think of a fiber. That fiber then wraps around some nucleosomes. And then those nucleosomes wrap together where it almost makes like another thicker strand where they start to coil to create a thicker fiber, which then winds within itself all the way around to make another fiber that then wraps and coils to make a chromosome. So if you were to think about a rope, a really thick gauge rope, and you were going to split it, it could be made of up to 10 fibers. And then you were to take one of those fibers and to split it into 10 fibers, and then can take one of that and to split it. That is a really good analogy of how your chromosomes are made. It's nothing but your DNA, just extremely and efficiently, tightly wound and compacted together. Because again, in every cell, in every part of your body, there are 23 chromosome pairs, 46 total, 23 from your mom, 23 from your dad, all of a complete copy of your DNA. It's pretty efficient. So we talked about how the 23rd pair is your sex chromosome. So males have 44 total autosomal chromosomes and 2 sex chromosomes. What makes males is the Y. If you have an XY for your last pair, both are being dominant, as you can tell because they're both capital, then you will be a male. Females have 44 autosomal chromosomes and 2 X for their sex chromosomes. So pretty much if you look at that, females can only produce females. They can either give the X or an X. It's the male who determines the sex of the offspring. If the male is fertilized with a sperm carrying the X chromosome, then it will be a daughter. If the offspring is fertilized with the sperm carrying the Y chromosome, then it will be a son. So here is a map of the human genome. The human genome is just a map of all 23 pairs, all 46 chromosomes together. Notice on every chromosome they do kind of have this X shape, but they do create bands. This is where the genes are located. And again, a gene is just made up of a section of DNA that codes for a specific trait. Here's a real image of a person's human genome. This was taken using an electron microscope, the most powerful microscope that we have. And you can see that as we order them, the chromosomes do get smaller. And then you have your very last pair, which are our sex chromosomes. And you can tell right here that this organism was a female because it's XX. Here's another organism. And you can tell that this one was a male because it's XY. Notice how much smaller the Y chromosome is. Okay, if you've heard of the genetic mutation of Down syndrome is when an organism is actually born and what is wrong is on this 22nd pair, that instead of only having 2 chromosomes, one from the mom, one from the dad, something happens and goes wrong and they get 3. So they might have gotten both moms and then one dad, or both dads and one mom. We're not really sure how, but that when you have 3 chromosomes on your 23rd pair, that's what causes Down syndrome. So let's talk about the actual definition, of a trait. A trait are the physical characteristics that are controlled by your genetic material. So if you have blue eyes, that is a trait. The gene that codes for your blue eyes is not. Okay, so the gene is what makes it happen. The trait is what we see. If somebody has curly hair, that is a trait. It's a physical characteristic that you can lay your eyes on. Genetic material of the organism are found in those rod-like structures that we said is a chromosome, which is located in the nucleus. So chromosomes, the actual definition, are made partly of long chains of molecules of DNA, which are then broken into sections called a gene. A gene is that segment of DNA that controls the information that governs that specific trait. So I might have the gene, I have the gene for blue eyes, but I do not have blue eyes. I have green. So even though I carry the blue eye gene, my trait is green eyes. So make sure you truly understand the difference between a gene and a trait. A gene is a section of your DNA. A trait is the physical characteristic. So let's talk about DNA. This is going to be new for you. You are going to learn about DNA next year in biology, so this is going to be very beneficial. DNA actually stands for the words deoxyribose nucleic acid. The DNA, deoxyribose nucleic acid. There happen to be 4 bases in all DNA, and which is pretty marveling when you think about it. The fact that our closest relative, the chimpanzees, they're 97% match our DNA. That 3% difference makes that huge change. And all that is off of 4 bases. So the 4 bases of DNA is adenine, thymine, cytosine, and guanine. We abbreviate those with an A, T, C, and G. So here's how it matches up. The adenine will only bind with a thymine, and thymine will only bind with an adenine. The cytosine and the guanine will only bind together. So you will never have an A and a C. You'll never have a T and a G. So just remember at, and then C and G kind of look the same. This is a great picture to show the levels of organization of DNA. And in fact, I think that was the last question. Yeah, it was. It was the last question on your benchmark that you took earlier this week. So you have your DNA bases. Notice how the A and the T fit together nicely, where the C and the G map like puzzle pieces. If you take a bunch of the bases together, that can make the double helix. A bunch of the double helix with a section makes a gene. All the genes together create the banding in a chromosome. And when you put all the banding together, that makes a chromosome. All 46 chromosomes make the human genome, and all of this is located in every single nucleus and every single cell, all trillion of them in your body. It's pretty amazing. So as you know from 6th grade and also from 7th, there are 2 types of reproduction for organisms. There's either sexual reproduction and asexual reproduction. Because we are talking about humans, we are mainly going to focus on sexual reproduction. So remember, sexual reproduction involves 2 parents who combine their genetic material to produce that new offspring. It does involve the egg cell from the female and the sperm cell from the male. And each combines half the number of chromosomes of that donor cell, the mother or the father. When the 2 come together, the egg and the sperm come together, that forms a zygote, which now has a full copy of all 46 chromosomes, or 23 pairs. Remember that sexual reproduction does create genetic diversity, right? So that means the offspring is going to be different from their parents. Even though you might look very similar to one, you're still 50% your mom and 50% your dad. So here's just another picture showing the sperm, which has 23 chromosomes from the male, the egg having 23 chromosomes from the female, Put them together, you now have your new offspring, which is a zygote. That zygote then will split, divide, and multiply until it becomes a new offspring. Here's another good visual representation to show you how it is diverse. Okay, so let's talk about this case. This was a 1 in a million. This was a biracial couple. The father was African American and the mother was Caucasian. They had twins. But what actually happened was one offspring was completely dark skin, dark hair, and dark eyes, where the other one was fair skin, blonde hair, and blue eyes. They are fraternal twins, born at the same time. It just happens to be that one was fertilized with an egg and a sperm that carried fair, and the other one was fertilized with a sperm that carried dark. So it's 1 in a million. They are still full blood siblings, but they look extremely different. Scientists actually believe that our skin color isn't just determined by one gene, but actually a combination of 7 different genes working together. And if you think about it, it kind of makes sense because there's not just one shade of white, one shade of black, one shade of tan. There's multiple different variations, and that's because there's multiple genes working together to create our skin color. So I'm going to go really quickly through this. You don't need to write any of this down, but we're just going to kind of review with Cookie and Cream. So here we have Cookie, he's our male, and Cream, she's our female. They're beautiful lizards. If we wanted to stop and look at some traits of them, notice how Cookie is green, and he does have these ridges on his back. Where Cream is blue, she does not have any ridges, but she has this bright banding on her tail. So if they decide that they're going to mate and have some offspring, they have peanut butter and jelly. Okay? Notice how peanut butter is a male, but he does have the blue skin from his mother, but he does not have the ridges from his father or the banding on the tail. Where jelly is a female, she also has the blue color from her mother, but she does have the ridges on her back and the banding on the tail. So even though they are full siblings, they do not look identical to each other or to one of the parents. So it's all about competition, right? In the wild, it's about survival of the fittest. The goal is to survive long enough to reproduce. Once this happens in the wild, you've been considered to have won the game of life. You passed on your genetic material. So peanut butter is competing with all the other males, lizards, and jelly is competing with all the other females to find their perfect mate and to pass on their genetic material. But let's also think about traits that can allow them to survive long enough to get to that point. Well, if jelly's back spikes keep her from being eaten by possums, but if peanut butter's lack of bright stripes keeps him from being noticed by owls, then maybe they both have a shot to live long enough to reproduce, and therefore they can die happy lizards. So let's talk about alleles. Most of the genes of a person in the chromosome come in pairs, which we call an allele. Alleles are different forms of a gene. So you can have a dominant allele or a recessive allele. So each gene is made up of 2 alleles. So you know when we write things for Punnett Squares, we're like capital R, little r. Each one, the capital R is an allele, the little r is an allele. Big R, little r together is a gene. The combination of the alleles of that organism is what helps to show the trait for the organism. So there are 2 kinds of alleles, and we talked about this. There's dominant and recessive. A dominant allele is one whose trait is always going to show. It's dominant. It's always going to be there. And we represent it with a capital letter. Recessive alleles are hidden or covered up, which means they are only going to show if there is no dominant present. So if there's a lack of a dominant allele, then recessive will show only if both are recessive, and we represent it with a lowercase letter. So let's talk about some traits. You're going to be doing this in a little bit. You're going to be doing a questionnaire on your own traits and then polling the class. Do you have attached or unattached earlobes? If you look here on the left, notice how his earlobe attaches to the base of his head. That is what we considered attached. Okay? This earlobe, see how it kind of it goes down and then goes back up and it attaches right here, we consider this to be unattached. So it has like that dangly little part. This is unattached, attached. There's a gene that controls that. So let's say the alleles for free earlobes, which is unattached, is dominant, capital F. And to have attached earlobes is recessive, little f. If a person inherits 2 dominant alleles, big F, for this trait, they're going to have free earlobes. If a person inherits 1 dominant, 1 recessive, which remember we call heterozygous, they're still going to have free earlobes because they still have this dominant trait. The only way that you can have attached earlobes is if both of the alleles are recessive. So just to make sure that you understand, this is an allele, capital F. This is an allele, lowercase f. Big F is a gene to have the trait of free earlobes. This is a gene to have the trait of free earlobes. And these two together are a gene to have the trait of attached earlobes. Okay, so I really hope we understand the difference between a trait, a gene, and an allele, because it can be extremely confusing. So just examples of other traits, because we are going to be getting into pedigrees and things like that. We have our human genome here. We have another example of how they can combine to make offspring, as well as what we're going to be focusing on, a pedigree to show a particular trait and how it's passed along, whether you're unaffected, affected. Okay? Now, he's an unaffected child because he has 2 capital letters. They're what we're called unaffected because they aren't affected by the gene, but they're still called carriers because they're carrying this little c, which is what is the trait that we might be tracing. So a person who carries the gene but doesn't show the gene, we call them a carrier. So an example would be, I'm a carrier for the blue-eyed gene. Both of my daughters have blue eyes, but I do not. But the only way they could have had blue eyes if it's a homozygous recessive. So I'm considered a carrier for the blue-eye gene, but I don't show it. So let's talk about the difference between genotype and phenotype. This was definitely a review from 7th grade, but it's so confusing and important that you understand. The genotype is the genetic alleles for the characteristic. It is on the inside, meaning I can't look at you and see your genotype. Okay, so an example of a genotype is big F, little f, little f. Those are genotypes. If I say, oh, you're homozygous recessive, you're homozygous dominant, you're heterozygous, those are genotypes. They're what codes for what's going to happen. Phenotype is the physical characteristic that is presented on that organism. It's what I can see with my eye. So I can look at you and I can see, oh, you have blue eyes. That is your phenotype. Now you might have little b, which would be your genotype, to give you the blue eyes, which is your phenotype. Okay? So if you can look at somebody and tell me whether they have attached earlobes or not, you're seeing their phenotype. If you're going to write it down, big F, little f, then that is their genotype. Okay? So brown eyes, blue eyes. What does it look like? Phenotype. What is it coded for? How do we write it down with the letters? Genotype. Make sure you have this down. This is definitely going to be a test question at the end of the unit. Okay, so we're just going to do a quick review over Mendel, because if you talk about genetics, you have to go into Gregor Mendel, which is considered the father of genetics. He was actually an Australian monk who did his work with pea plants, and he laid the foundation for genetics. He is considered the father of genetics, So his experiment involved testing 7 traits, okay? The color of the flowers, the positioning of them, the pea color, the pea shape, the pod color, the pod shape, and the height of the plant. Notice he has put all the dominant ones on top. So purple is dominant to white. Axil, which is along the side, is dominant to aerial, which is across the top, or terminal across the top. Yellow is dominant to green. Round is dominant to wrinkled. Green is dominant to yellow. Inflated is dominant to constricted. And tall is dominant to short. So Punnett squares. Love Punnett squares. Remember, a Punnett square is a diagram that is used to help you figure out the probability of that trait being passed on. We use uppercase to represent dominant and lowercase to represent recessive. Okay, right? I'll review. So here we go, a Punnett square. Remember, you are always going to put the female's genotype down their left and the male's genotype across the right. Okay, so if you look at this example, Mendel crossed this plant with this one. Notice this is tall and this one is short. So that happens to be the trait that we are passing. So we have it with a big T. So the female was obviously homozygous dominant, or the male is homozygous recessive. So we can automatically see that tall is dominant to recessive. If you were to cross these, then does this mean you're going to have 4 offspring? No. This just means of this one offspring, it has 100% chance of being tall. So if you cross these plants, every single time, their offspring is going to be tall, but it's going to be heterozygous. So again, we call this a carrier, right? They carry the small gene, even though they themselves are not small, but they're tall. Okay, so let's talk about the difference between a purebred versus a hybrid, because that's going to go into our Punnett square. Purebred is a terminology to mean homozygous, which again we know means same. There is only one type of allele. Big B, little b, homozygous dominant, homozygous recessive. You're a purebred. Hybrid, which means heterozygous, because you're a mixture. You're a bunch of different things, which is big B, little b. Another word for hybrid heterozygous can also be carrier. So remember on the Punnett square, you're just going to do a tic-tac-toe. You don't have to draw the outside boxes. I don't because I'm lazy, but you can. Females go down the left, males go across the right, and you just drag and move over, okay, to create the probability of one offspring. And then you can put them in a percent or a ratio. Okay, so I want you to do this, and I'll check it. Tall and short, which is dominant, which is recessive. Easy, you should be able to tell. And we're not going to go into controversial genetics or any of this other stuff. Okay, so that's pretty much, we're going to go back here, and this is where we're going to lead off. You are then going to trait your own genetic traits, do a group. You're going to create a bar graph in your journal with color, and then if you get finished, you can do a fun worksheet on Monster Inc. genetics. Okay, so I hope this has kind of been a great refresher for some of you. You've taken really good notes because you are going to need them for the test and this next assignment. I'll see you Monday.